So welcome everyone to those who have just joined. I think we will slowly start. So my name is Alexandra Haulinsky. I'm a scientific training officer at EMBL-EBI, and I welcome everyone who joined today for this webinar. Before we move on to the webinar, I will very quickly run you through some instructions for today. So all microphones will be kept muted throughout the webinar, and if you have a question, please type it into the Zoom Q&A box. You can even upvote questions that you like. Oops. And we will try to answer as many questions as possible at the very end of the webinar. Please complete the feedback survey, which will be launched at the end of the webinar, because this will be very helpful for us to plan future webinars. And it's also important to let you know that the webinar will be recorded and added to EMBL-EBI's on-demand trainings, listings, and YouTube. Next slide, please. So today's webinar is the third one, sorry, of our webinars of our webinar series on open access and open science. And there will be one more next Wednesday, and we will then have also a panel discussion next Friday, which will also mark the end of Open Access Week next week. In today's webinar, we will focus on finding and accessing open data. And with this, I would like to introduce you to the speakers of today. So the first speaker will be Anna Swan. Anna Swan is a scientific training officer at EMBL-EBI, and she's focused on e-learning. And in her part, she will today she will give an overview of the kind of data which is accessible through EMBL-EBI. She will also demonstrate EBI search for finding data at EMBL-EBI, and will introduce the concept of programmatic access. I would then also like to introduce you to Yuge Sarkans. Yuge Sarkans is a team leader for the functional genomics development team at EMBL-EBI, and he will focus today on BioStudies, which is a database that holds descriptions of biological studies and links to data in EMBL-EBI resources, but also beyond. I'm very happy that both speakers are here today, and with this, I will hand over to Anna.
Thanks, Alex. And thank you to everyone who is joining us today. So Alex said, I'm going to talk a bit, I've got ahead of myself already. I'm going to talk a bit about what data you can access from EMBL-EBI, what kind of resources we have available, we provide, and some different ways that you can actually access that data, so that hopefully you can go away at the end and try things out for yourself. So as a rhetorical question, how do you currently search for data? How would you go about searching for biological data if you were to open up a browser now and have a search for something? Perhaps you would just go to Google straight away. Maybe you would try and look for literature. You might go to Google Scholar, PubMed, Europe PMC, or you may go to NCBI or, in fact, EBI itself. So what I'm going to be talking about is our website that you can see down there. And in terms of data that you can actually get from the EBI, it's often a lot broader than many people initially realise. Often people know about resources such as Ensemble, Uniprot, they know they're from EBI, but there's actually a lot more available. So there are, for example, literature-related databases, so Europe PMC that I've already mentioned, and Yuge's going to talk about BioStudies in his section of today. There are, obviously, as I mentioned, gene information available via Ensemble. There's expression data. There's protein structure data through PDBE. There's protein-protein interaction data, mathematical models through biomodels, going right the way along this path here to drug discovery data through open targets. And I'd really encourage you to go along and have a look at this EBI/services page, because there's a list there of all the different resources and tools that the EBI does have on offer. And it can just be very interesting to take a look to understand the breadth of data and tools that are actually available. Just to mention at this point, these slides will be made available along with the recording of today. So don't worry about scribbling down the URLs that I've got in these slides. We'll make sure that you get a copy of these slides tomorrow. So if you do go to that services page on our website, this is what you'll see. You'll see a list of tools and different data resources, and this is a long list. So do check that out. You can reduce it a little bit by searching for something in particular that you're looking for. But one thing I did want to highlight from this page is this right down at the bottom. I've cut it off here. It's our principles of service provision that I've expanded on the right here, and right at the top of that is what we're kind of focusing on this month ahead of Open Access Week, being open. And it's really one of the main principles of the EBI, to make our data and our tools freely available to anyone who wants to use them via our various different resource sites. There is an exception to that, which is mentioned there, where there's potentially identifiable human genetic information, patient-related data. So, for example, through the European Genome Phenome Archive, the EGA, that is restricted access. So, for example, with the EGA, you can go and you can look at the different types of data sets that are available. If you find something that you are interested in studying, you can apply to use it. So it's not restricted in that you as an individual can't access it, but it is restricted that you have to give your reasoning and explain your research so that you can access it. And we do cover lots of different species. So not just human, not just mouse. There's all sorts of plants, insects. There's something here that I'm not quite sure what it is, but there's lots of different things. So it's certainly not restricted by species. One of the main advantages of accessing data through the EBI is that the resources aren't all standalone. Often they are integrated and work alongside other resources. So perhaps you're in a page from one resource, but there's a link taking you through to more information about that gene or protein through another resource. So it allows you to kind of follow a pipeline to access more and more information about the thing you're actually looking for. So it's providing you extra information, extra steps that perhaps you may not necessarily anticipate when you first come to the resource. And whilst we are the European Bioinformatics Institute, our data resource teams do really collaborate with lots of other teams all around the world, and our data is accessed by everyone all around the world. And these collaborations are really important in order to make our data, as it says, as open as possible and as sustainable as possible, so that we can keep up to date and keep supplying that data. And in terms of collaborating, some of the resources are mirrored in other resources. So we often get the question of, what's the difference between PDBE and PDBJ? Well, actually, they're pretty much the same data, but what differs is the web interface and sometimes the tools and the algorithms there that said that are available. So it would be fine if you're accessing that data through one of the other sort of similar mirrored sites, but then there may be a different tool. You may prefer one of the interfaces over another. So that option is there for you. As I mentioned, we are used very much all around the world, not just within Europe. I took this in the morning in the UK, so you can see everything's a bit more focused in the middle here, but if I'd waited until a bit later in the day, you'd find lots of people waking up over in the West here. So don't feel restricted if you're not in Europe. The data is very much there for you to use wherever you are. But having said all this, it can be quite confusing. If you go to that services page, you're not quite sure what you're looking for. You know the kind of information you want to find, but you're not quite sure how to find it, which resource to go to, what tool to use. This is why we have EMBL-EBI search. This is why we have EBI search ready for you to use. If you go to ebi.ac.uk, the site at the top here, and this is the front page of our website, you will find right at the top there is this search button, search box. And as it says there, find a gene, protein, or chemical, you can put whatever you're looking for into there, and it will return all the information that you could possibly want from the EBI. So here is an example. I put in my query, and I will jump, in fact, to the website and show you what that looks like live. So at the top here, this is our live site. At the top here, we have, No, let's go for, there we go. Put our search into there, hit search. That's all I've put in, nothing else. And you will see down the left-hand side, there's all sorts of different kind of results. So it depends very much on what you're looking for, but there's a huge amount of information there for you. But what's really valuable is that it also says the source. So that's the resource that it's coming from. So for example, this is coming from Uniprot. I'm interested to see a bit more about this. I can click straight through and see that page on the Uniprot site. And that's one of its main advantages. I didn't need to know I was looking for Uniprot originally. I just could come here and have a look through the different types of data that are returned, and then just go straight through to that example within the relevant resource. So that was a really quick demo, but that was really what I wanted to show you, because it's right there at the top of our page for you to go and try out for yourself. And I really recommend trying out and having a test around, play around, see what you can find, perhaps what you can find that you weren't expecting to find, and test out the various resources in that way, rather than just trying to jump into an individual resource that perhaps it may not be quite what you're looking for. So jumping back to the slides. Alex mentioned about talking about the concept of programmatic access. So EBI search and going straight to the resources is great if you're looking for one or two bits of information. But what happens if you've got a whole list of genes you want to find out information for, or you're trying to bring information into a pipeline that you're running? And this is when programmatic access really becomes so important. When you go back to the services page that I showed before, you will see that under all the resources that have it is this little web API, and that says this resource can be accessed via an API. And at that point, what I would recommend is going to the resources website and actually looking at, usually there's a tab that says API, having a bit more information there available for you. But what is also really useful, and I will share this link during the session because I think this is very useful for you. There are many trainings available on using the various resources APIs. So for example, on the right hand side here, we've got the a screenshot from the Ensemble API page, and this is really useful, provides you all the details of how to use it. But what you may actually be more interested in is they actually provide a Jupyter notebook. So if you're getting started, perhaps using R as the example is here, you can use that notebook to get started and actually learn how to use the API rather than having to work straight from the documentation. And then on the left here, this is you can't really see it, but it's a screenshot from the Magnify API webinar. So this is a webinar where someone from the Magnify team took everyone through how to use the API, and that recording is there available for you to work through, so that you can kind of take your time working through that in step with the webinar. And once I've stopped talking, I will share this link into the chat with you so that you can take a look at that. But there's lots available on different resources. And as I said, you can have these slides as well, so you can have that link at that point. So hopefully that gives you an idea about why and how to get started with the programmatic access. It is different for lots of different resources, so we're not going to go into great detail, but there is a lot of information, and hopefully you can understand that it may be really useful for you if you are trying to access large amounts of data. So the last thing I wanted to mention was just about how to cite data from these various resources. So this is a general overview of how you may want to cite the data. Obviously, it's a little bit different to a paper, but it's not massively different. But also often you need to check the resource database, as they tend to have a paper as well that they'd like you to cite. But the one thing I wanted to really highlight was to watch out for releases or versions, because information in databases can change. So it isn't necessarily enough to just have the URL of the page of the data that you were looking at. You need to know when it was, and you need to to check the version of as well, so that if you need to, you can go back to a previous version, and it will also explain any changes that you might see. So that's a really important bit, just to notice that you do need to keep track of that. Okay, so I think that is all from me, and at that point I'm going to hand over to you, Liz. Thank you.
Thank you, Anna, and thank you everybody for logging in. Let me just share my screen. So let's talk about the BioStudies database. So that's one of the several numerous resources that EBI provides. And so before I start defining what BioStudies does, just briefly reflect on sort of the data heterogeneity problem in life sciences. Of course, as Anna mentioned, there are many types of structured data for sequence, for proteomics, for protein structure, and so on. And there are specialized resources at EBI and in other places that provide and deal with those data types. But then there is also more and more what are sometimes called multi-omics studies, where in the same study generated different data types that were deposited into several community databases, and then it becomes a bit harder to find everything related to that study. But there is also what is sometimes called unstructured data. I don't particularly like the term unstructured data. There is hopefully always some structure. It's just that structure is not defined in advance, so to say. So what happens to that data? And then there is also data that clearly has structure, but the community standards and resources for sharing that structure those are not yet present. And light microscopy data could be one of the examples where the data sharing approaches are still developing right now. And then from slightly different angle, well, there are publications with supplementary data attached, and then typically you access in a variety of ways from publishers' websites, and that's okay if you want to access data for 1 or 3 papers. But what if you want to gather data from hundreds of different papers? It then becomes quite complicated. So these are some of the problems that the BioStudies database tries to help with. And let's look at one concrete example. So there is a paper related to SARS-CoV-2, so very topical paper. And so we might want to ask questions. So what the data are associated with this paper? In which community databases are those data sets? Are all the associations between the paper and the data set of the same nature? And what else is attached to this paper? Are there any supplementary materials? So now if we read the paper, then, well, I've copied out some paragraphs from that particular paper. Well, then you can see that these links to data sets are perhaps a little bit hidden. So here there are a few links to the protein structures, and then if we read on, so there is a link to supplementary figures. So it's perhaps not always very easy to figure out what data is there. And then, well, BioStudies. So now I come to the definition of what BioStudies is. And the aim of BioStudies. So the aim is to facilitate transparency and reproducibility of research by aggregating all the outputs of a study, what we sometimes call a data package, in a single place. And the diagram on the right-hand side sort of tries to graphically represent that. So the data package really has the links to EBI resources or external resources, then what we could call orphan data, some PDFs, some Excel files, things like that. And then, of course, each data package needs some metadata. Otherwise, it's not possible to interpret what that data package means. And a link back to publication if one exists in Europe PMC database. So a single study then is typically associated with a single publication, but that's not always the case. It may contain data from a unit of work in a larger project. So now if we return to the same paper, this is the representation of that paper in the Europe PMC database, which is another resource at EBI devoted to literature management. So I can recommend for all your scientific paper needs, please use Europe PMC. It's great. And then the paper entry in Europe PMC links to the data set in BioStudies. So this is now a screenshot from the BioStudies database, which has the paper title, author's abstract, so metadata about the paper, as well as that supplementary data file and the PDB links here. So that's one example how data is structured in BioStudies. Before I go into some more sort of practical details about the use of BioStudies, let me just again briefly return to sort of how BioStudies is used across the research life cycle. Was the paper things happened before the paper was written? There was a research project was ongoing, and we collaborate with the BioStudies on a number of projects during the data generation stage, where we help with data sharing and eventually publishing those data sets. And another example is supporting an emerging community. For example, for protein design community, standards do not yet exist. So we try to help with BioStudies that community. But then what happens after the data has been generated? So then there is a manuscript preparation stage where again BioStudies can be used to directly deposit the data sets that do not have a dedicated place in the larger life sciences data ecosystem. I have mentioned the Bioimage Archive and their Express here. I will return briefly to those resources. And then after the publication, as I already mentioned, BioStudies takes the data packages automatically generated by the Europe PMC team, and we make those available. And we also collaborate with a project called Source Data, which again I will explain in a bit more detail. And finally, there are cases when there are data resources that cease to exist for some reason, and then BioStudies can be used to as a home for data sets from retired projects. Express appears twice in this diagram. Again, I will later explain what that means. But more generally, so the sooner data sets are structured and the data sharing problem is thought about, the better it is for reproducibility, for open science, for fair data. So all these concepts, which are, of course, these days more and more talked about, really. So okay, so that's all quite abstract. So let's now just briefly look at data access in BioStudies. These will be quite practical screenshots. Not too many of those. I think it's a good data resource should be sort of obvious how to query and how to use that. So I don't think I need to spend too much time on that. So if you go to EBI/BioStudies, you will see in the browse view we have almost 2 million different data sets. but most of these, more than 99%, have been actually automatically ingested from the Europe EMC by automatically mining papers for accession numbers and packaging together with supplementary data files, and then giving those data packages to the BioStudies database. So what else? Well, there's a variety of ways to sort data sets. And then here on the left, so we have a concept of collection in BioStudies, which is a group of data sets acquired sort of from the same pipeline. So Europe PMC, as you see here, that has this large, the largest number of hits, and all the other data collections are much smaller. But I will talk about some of these in a bit more detail. And then there are other facets that can be used to narrow down your set of results by the release date here. And then if you scroll on, for example, if you would like to find all the data sets with a PDB entry attached, then it's possible to do that just by a single click. And also filtering by file types that have been attached to a particular data set. Again, this is perhaps not very useful for PDF, but if or very generic file types. But there are some file types which are specific for a particular context. So, and then if you click on a particular data set, then that's how a data set is rendered in BioStudies. On the left-hand side, you have a metadata, so study annotation, and on the right-hand side, you have a list of files and a list of links to components of data in some other databases. In this particular case, there are not many of these, so 5 files and 16 links, but there are also means to deal with much larger numbers of files and links, if that is what a particular data set has. So in this example, the metadata is quite minimal, but the BioStudies model is quite flexible, so it can accommodate also much more complicated metadata structure. So in this example, there is an imaging study, and for imaging, the metadata happens to be much more detailed. So the imaging study, the metadata fields include imaging methods, phenotype, quality control description, and some other things. And here also you can see that there are many more files, data files here available for access. And another example, even more sort of complicated metadata. So I already mentioned the source data. So these are our collaborators. So the idea of the source data project is essentially that it's not good enough to provide a figure in a paper just as a set of pixels, and that it's much better if also the data, that let's say the data matrix behind a particular graph is made available, as well as the entities that are shown on the particular figure are actually spelled out and annotated. And this is what happens here. So we have a paper which has a number of figures, and for each of those figures, the data files that were used to generate those figures are provided, and they are available via BioStudies. These are the links to the actual figures on the source data site, and these are the tables that sort of describe the biological entities that are present in the figures. So actin, which is a protein, lymph node, which is a tissue, and so on and so forth, linked to the appropriate repositories if relevant, and all that information is searchable in BioStudies. Okay, so let's briefly talk about search now. well, search should be very intuitive to use. Hopefully, it is. So one thing to expand on is that the search is, in this case, we have found also more specific terms. So it's not only hyperplasia, but also these different types of hyperplasia. So how does the system know that this hierarchy of terms? So what we use under the hood is ontology. So it's a hierarchy of terms. A particular ontology that we use is called experimental factor ontology, EFO, which is another resource that is being built and maintained at the EBI, and which provides a sort of a systematic way of describing experimental variables that are used in various EBI databases. So it is possible to combine search terms. So if you want to find everything about lamprey guts, then you can use this particular query. Again, some coloring here. This C. lamprey happens to be a synonym of lamprey in the experimental factor ontology. So the BioStudies knows about that. Yes, it's possible to use these keywords in capital letters and brackets to combine search terms, as in this particular example, or can be skipped if you just put in a number of terms. Then it will be assumed that you want to find all the data sets related that would have term 1 or term 2 or term 3. So if you want to find a phrase, then you need to put that phrase in quotes, as in many search systems. And then talking a bit more about facets. So depending on the collection, there may be different set of facets available. For example, for the Europe, for the data sets imported from the Europe PMC dataset database, we have a funding agency faceting available. This is actually not very clean data, but it is a hard problem to filter, to find all the papers, all the data sets associated with the funding of a particular agency. But that is where we are trying to do that. And for other collections, there will be some other search facets available. So that's all about finding data sets. And then what about downloading the actual data files? So in this example, we have a much larger data set than what we saw before. We have almost 35,000 data files in this imaging experiment. So you can use this file panel for searching for particular data files, and then it's possible to select only the data files that you would like to download. And then there is a number of download methods available. You can download everything as a zip file, but again, for a very large imaging data set, that will perhaps not be feasible. So the system also can generate an FTP script or an Aspera script that you can then run on your system in order to download a subset of the larger study or everything, if that's what you need. Okay, so I think that's about some practicalities of using BioStudies, and I will wrap up by talking what I promised at the beginning, to talk about the Bioimage Archive and DERA-Express. So Bioimage Archive is quite a new public resource at EBI. Matthew Hartley is running that resource. It was launched a couple of years ago, and the idea is here to store and distribute biological image data sets that are useful to life sciences researchers. So these are the objectives. Again, reproducibility, providing reference biological imaging data sets that can be used in subsequent studies, also allowing new scientific discoveries, perhaps by combining different data sets, and also accelerating research in bioimaging informatics by providing a large number of data sets that can then be used to fine-tune your image machine learning algorithms, for example. So please feel free to explore this EBI/Bioimage Archive website. And if you click on Browse, you will notice that the interface looks very similar to the BioStudies interface. And well, the reason is that actually under the hood, at the moment, the Bioimage Archive is using the BioStudies engine, and essentially Bioimage Archive is a collection of data in BioStudies at the moment. So all the functionality that is generally available in BioStudies is also here. So you can search for image data sets. We index also the Empire data resource, which is an electron microscopy raw images, raw electron microscopy images. So that's another resource at EBI. As well as the data sets that are directly in the BioStudies. And this is just another example of an imaging data set. So this has in total 100,000 files, and it's about cellular phenotyping study. So it's possible here to try and find sort of narrow down all the set of images that you are looking at or you want to download. And another example of a SARS-CoV-2 study, again, large number of files, and we have for a few imaging data sets, we have generated thumbnails that are perhaps somewhat useful sort of for visual exploration of data sets. In this case, we have references to compound information. So that was about Bioimage Archive, and then finally about ArrayExpress. To explain why I mentioned ArrayExpress and why it was mentioned in two places in that diagram. So ArrayExpress is being migrated to the BioStudies backend. It's coming very soon. ArrayExpress is a database that has been around for around 20 years already. It's mostly about gene expression data. So we have around 75,000 gene expression data sets available from ArrayExpress. And what you see here is the ArrayExpress user interface that you can use for downloading data, for finding and downloading gene expression data sets. So this is, we noticed a few years ago that we do not need to maintain a separate data access interface for ArrayExpress, and that BioStudies can also be used for the same purpose. So here we have a BioStudies view where we are looking at all the ArrayExpress studies. So it's currently just a small number because the migration is in process. Yes, but this is how essentially in future, when you go to EBI/ArrayExpress, you will be automatically redirected to this BioStudies collection. And then just again looking at a single data set, this is how the single data set looks in the ArrayExpress specific user interface, and this is how it will look like in the BioStudies. So we are retaining the structure that is necessary to interpret a gene expression experiment, things like descriptions of your samples, experimental factors, and so on. And we are in BioStudies, we can provide the same that information, and keeping the structure essentially. Finishing with the acknowledgement slide for the BioStudies team for funding and the source data collaborators. Thank you. Back to you, Anna.
Yeah, thank you very much, Lucas and Anna, for the great talk. So before we move on to the question and answer session, I would very quickly like to remind everyone that we have upcoming webinars next week. There will be one on Wednesday on publications and preprints as part of this webinar series on open access. And there will be another one delivered by Uniprot, where Uniprot presents its new webpage, which will be released soon. So that might be also of interest for some of you. All our webinars are accessible on our EBI training webpage. And as I've mentioned earlier, we also make recordings of all of our webinars, and you can access the recordings after the webinars have taken place also on our EBI training webpage. I would like to remind you also that there will be a feedback survey launched at the very end of this webinar. So please answer this feedback survey, as this will be very helpful for us to develop future webinars. And with this, I think we can move on to the question and answer session. So if you still have a question, please add this to the question and answer box now. And we already have 3. So the first question, I will go simply from top to down. Do EMBL and NCBI have or share the exactly same gene expression, so transcriptome data sets? And there is a second part, does EMBL have a tool similar to NCBI Geo2R?
Yeah, I can answer that. So we do not share exactly the same transcriptome data sets. There was never a collaboration like for sequence data exchange. That was not set up for exchange between ArrayExpress and the Geo resource. And ArrayExpress for a long time also imported data from Geo, but that was stopped around 5 years ago already, because it was not seen as providing enough value to the users. There is a package in Bioconductor that can be used that helps essentially with dealing with gene expression data sets in ArrayExpress, which essentially understands the MHTab format, which is the format that underlies gene expression metadata exchange.
Okay, thank you. And I think there is another one for you, Lucas. Could one use BioStudies to combine human transcriptomics and proteomics data sets to generate some sort of proteogenomics data?
Well, BioStudies helps with finding data sets. It will not help with the actual problem of combining data. So in general, all the data that is in BioStudies is available under the EBI terms of use, which in spirit are very close to CC0 license. So the data, well, essentially feel free to reuse the data in what that you find in BioStudies and really for any purpose. But yeah, I cannot sort of for the specific problem of I think human transcriptomics in the first place, there is also the implications on privacy. That's human data, right? So it's likely that many human transcriptomics data sets live in the EGA. So that's the specific EBI resource that Anna mentioned that has special rules on accessing, which is not where the data sets are not just freely available for everybody.
Okay, thank you. Another one, will accession numbers for ArrayExpress studies change if it is migrated to BioStudies?
No, the accession numbers will stay the same. I think that's one of the good principles that we have always tried to observe, that accession numbers should never change, and we will also maintain the URLs that currently, for example, EBI/ArrayExpress/ specific accession number. So that will still work, only it will go to a different place. It will show up as a different web page. But yes, we are not changing accession number.
Yeah, and there's another question referring to ArrayExpress. Since ArrayExpress is being migrated to BioStudies, which database would you suggest to use for data downloading for now?
For now, please continue using RExpress because the BioStudies does not have the full set yet. Though actually we are also very interested for the data sets that are already in both places. If you have feedback on how we represent the data sets in BioStudies, we would be very happy to receive that feedback.
And I think the last one is an extension or an addition to the question which you already covered on the proteogenomics. So to combine human transcriptomics and proteomics data sets within the context of SAAVs.
I have to say I do not know what SAAVs is in this case.
Yeah, me neither. So unfortunately, if maybe the person can define what SAAVs means. Single amino acid variants.
Yeah, so I think that is a very specific question. I think I would say so. Proteomics data sets are in PRIDE, right? So perhaps if you write to the PRIDE help desk as well as to the RExpress help desk, then we will. I do not have access to the PRIDE help desk, but I do to the RExpress help desk, and we can follow this up offline. Yes, I do not have an answer right now.
Yeah. Well, so Anna, do you want to?
I was just going to say that's something to highlight as well. That all of these resources do have help desks with the people there to help you, and they're very much available for that purpose. So do get in contact if you need some.
Absolutely. And I think this was the last question. So I would like to thank all attendees of this webinar for the great questions. And of course, I would like to thank the speakers for being here, for delivering great insight, and also for answering all the questions. And yeah, I hope to see some of the attendees next week again and for the next webinar in the series. And thank you and goodbye.